The topic of this lecture is gene expression. Now here's something you've seen before, the central dogma of biology that shows the relationship between DNA, RNA, and protein. And remember that DNA represents information inside of the cell, whereas proteins represent function inside the cell. So what is the information in DNA? It's the information for making all of these different proteins. So an important thing to define carefully here is the idea of gene expression. So we can complete this sentence by saying that a gene is expressed when the information in that gene is actually being transcribed and translated so that the protein that it encodes is actually present in the cell. So a gene is expressed when the DNA info is transcribed and translated to produce a protein. All right, now I think a good analogy for gene expression is this, a little Swiss Army knife. Now, inside the Swiss Army knife, there are many different functions, right? There's a corkscrew and a screwdriver. Here we have another screwdriver and a bottle opener. We have a can opener. And of course, we also have a knife on here as well. Now, there's a reason that we don't carry the Swiss Army knife around in our pocket like this. I mean, besides the fact that it would be very painful, we don't need all of these functions all out ready to go at any moment, right? So the way we do it is we tuck them all away, and that we only pull out those functions when they're really needed, and then we have them there. But most of the time they are sort of stored away inside this object. Now we can think of this as an analogy for a bacteria. You know, a bacteria might have thousands of genes, all that have important functions, but those functions are not needed all the time. So most of the time those genes are not being expressed. The information is there in the DNA, but the functions that are encoded are not actually present inside the cell. But when the conditions of the cell change, and you do need a certain function, it's there, ready to go, ready to be expressed. So our example of gene expression is going to be a region of the DNA referred to as the Lac Operon. And this is an important term to define. An operon is a group of genes that have a related function. Genes with related function, which are all localized on the DNA together on the chromosome, and more importantly, their regulation or their gene expression is controlled together. So genes with related function and gene expression control in common. So they're all maybe part of the same biochemical pathway, as in the example of the Lac Operon, and they're regulated together. So the Lac Operon is important because it really is the first really well understood example of how gene expression is controlled. This is unique to prokaryotes. You don't really find operons like this in eukaryotic genomes. And also everything that we're going to diagram and discuss is also covered in the textbook in figures 17.5 through 17.12. Okay, now before we start actually diagramming DNA, I like to think about what I like to call the logic of the Lac Operon, why it makes sense for the bacteria to regulate these genes in the way that they do. So, first of all, lac refers to this molecule, lactose, which is a sugar molecule. It's actually made up of two 6-carbon sugars, a glucose and a galactose. And they're held together here by a glycosidic bond. Now below here are listed 3 points that are important things to keep in mind as you study and learn about the Lac Operon. So the first one is that this molecule, lactose, can be used as an energy source for the cell, but it's not always present. Right? A bacteria can go its entire existence without ever encountering a lactose molecule. However, if it does, there is a lot of energy stored in this molecule, and it might be useful for the bacteria to use that as an energy source. So, number 2, the genes encoding the enzymes that metabolize lactose are only needed when lactose is present. Right? It would be a waste of energy for the cell to make those enzymes all the time even when lactose isn't around, if the only function of those enzymes is to break down this molecule. So this is important from an efficiency standpoint for the cell. All right, now the third point is something that we'll come back to towards the end, and that is that if there are multiple sugars present, if lactose and glucose are both present, then glucose is the preferred energy source. And that makes sense too, also from an efficiency standpoint, because the cell always has the enzymes required for breaking down glucose. Those are the enzymes in the glycolysis pathway. So if glucose and lactose are present, it doesn't really make sense for the cell to express a whole bunch of different enzymes and to catalyze a whole bunch of reactions to break down the lactose when glucose is already present. It's an easier sugar to access for the cells. So these 3 points really make up what I like to think of as the logic of the Lac Operon, and we'll come back to these as we think about the way gene expression works in this case. Okay, next we're going to diagram the Lac Operon, so give yourself plenty of space here, and I'm going to start off by just drawing some double-stranded DNA, just a part of the bacterial chromosome. And right in the middle here, I'm going to put a little bent over arrow, and this represents the place where transcription starts. So this is the start of transcription. It occurs right here. Now, downstream of that, there are 3 genes that are going to be interesting to us. The first one is called LAC-Z. The next one is called LAC-Y. And then there's another one called LAC-A. So these are all genes that have a related function. They're all required for lactose metabolism. Required to metabolize lactose. So remember, an operon is described as a bunch of genes that have a related function. And so these are all, that is their common function. Now what's sort of interesting about this, and this only happens in prokaryotes, is that these 3 different genes are actually transcribed all together. So transcription produces a single RNA molecule. It begins right here at the start of transcription. There's the 5' end, and so we make this one RNA molecule that contains the information for actually making 3 different proteins. And for example, there might be a start codon here for the LAC-Z gene. There's another start codon, AUG, here for the LAC-Y gene. Another start codon here, AUG, for the LAC-A gene. And these are all going to be translated by ribosomes separately. So if this part gets translated, you end up making an enzyme. Try and draw the 3-dimensional structure here. An enzyme called beta-galactosidase. We'll just abbreviate it beta-gal. If this part of the RNA gets translated, then that produces actually an interesting integral membrane protein called the lactose permease. And this allows lactose molecules to move across the membrane into the cell. And then there's another protein here. We're not going to worry too much about its structure or function, but yet another protein involved in lactose metabolism here, encoded by this gene LAC-A. All right, so here we have 3 genes, LAC-Z, LAC-Y, and LAC-A. They can be transcribed to make an RNA molecule, and then that can get translated to make different proteins. One from LAC-Z, one from LAC-Y, and one from LAC-A. Now, this is the one we're really going to be focusing on most, not just in lecture, but also in the lab that's associated with this material. So the LAC-Z gene, when it's transcribed and translated, produces this enzyme called beta-galactosidase. And what beta-galactosidase does is it actually catalyzes the splitting of those 2 sugars. So if you have a lactose molecule as a glucose and a galactose held together by a glycosidic bond, what this beta-galactosidase enzyme does is it breaks that bond and it frees up the glucose and the galactose molecules. Okay, now we're not done diagramming everything because we have to include the regulatory DNA sequences over here. And there's 2 of them that are going to be important to us. The first one is called LAC-P, and this is also referred to as the promoter. And then this one here is LAC-O, also referred to as the operator. Okay, now we're going to deal with the operator in just a minute, but first we're going to start off thinking about what happens here at the promoter region. Okay, so what happens to initiate the transcription of all these genes? Well, that, of course, involves the enzyme RNA polymerase. So let's draw an RNA polymerase here. So for RNA polymerase to transcribe, it has to get here to this point, this where transcription starts, represented by this little arrow. So RNA polymerase begins by binding onto this promoter region, Lac P. So here's RNA polymerase binding onto Lac P, and then it has to move through this region called the operator to get to this point where transcription starts. And if it can do that, then it's going to begin making this RNA molecule here, transcribing using one of these DNA strands as a template to produce this RNA. Now, just for accuracy, the RNA polymerase doesn't bind onto the promoter by itself. There's also a protein called the sigma factor that binds on there as well. But the important thing is that RNA polymerase starts here at the promoter, it has to move through this operator region, and then transcription can begin. Now we'll come back to that diagram too, but now we're going to think a little bit about the dynamics of expression. So here in this graph, we're looking at the expression of the LAC-Z gene. Now remember, the LAC-Z gene product is a protein called beta-galactosidase that I drew on the other page here, beta-gal. So what we're looking at here is the concentration of that protein inside the cell and how it changes over time. So we're going to start with no lactose present. So if there's no lactose present, there are very, very low levels of this beta-galactosidase gene, almost down to nothing. Now let's say at this point we're going to add lactose. So you can do this in a lab, and in fact you will do this in the lab, where you will control when the bacteria get the particular sugar. So we're going to add lactose at this point. A little while later, what we'll see is that the expression of this gene increases, and we get an increase in beta-galactosidase enzyme. And eventually that will level off. Okay, so what we've had here, we add lactose, then there's a little lag, and then the gene is expressed. Now, we can also think of what would happen if we remove lactose. Or perhaps if the cell consumes all of the lactose that's present. So we remove lactose here, and after a short lag, then the beta-galactosidase levels are going to fall back down pretty much to 0. All right, so what this graph is really showing is that lactose is, or beta-galactosidase, this LAC-Z gene, is expressed in response to lactose. And so what we're really going to think about is this question down here: how is LAC-Z expression regulated by the presence or absence of lactose? Oh, and I should say too, comparing this graph, it's a good time to think back to our steps here that talk about the logic of the Lac Operon. Remember that lactose is a good energy source, but it's not always present. And so the beta-galactosidase gene is a good example of an enzyme that's only needed when lactose is present. And so, as the graph shows, once the lactose is present, or before lactose is present, you have very low levels of the gene. Once you get addition of lactose, then you get expression of the LAC-Z gene. That's what's going on here during this increase. And here, the expression gets turned off. So now we're going to think about how lactose controls the expression of beta-galactosidase. And this is an example of what in general can be referred to as negative control. Okay, so I'm going to draw just a small part of that bigger diagram that you saw before. Here's the start of transcription. I'm going to put it over here, so this would be LAC-Z off this direction. And what we're really going to focus on now is the regulatory regions. Here's Lac-O, the operator, and over here is Lac-P. the promoter. Okay, now there is another protein we haven't diagrammed or discussed yet, which is referred to as the repressor protein. Now the repressor protein is able to bind onto Lac O. It can recognize and bind to this specific DNA sequence here. So here I've shown the repressor protein either off the DNA and the cytoplasm or bound on to this region here called the operator. All right. So what causes and the important thing is when the repressor is bound on here, it prevents RNA polymerase, this other important enzyme here. It physically impedes the RNA polymerase from making it here to the start of transcription. So it really is just blocking the way there. So as long as the repressor is bound on here to the operator, you will not get expression of LAC-Z. All right, so how is this repressor controlled? Well, it has two conformational states. In one of those conformational states, it will bind to the repressor, or it will bind to the operator, and in the other conformational state, it will release the operator and go off into the cytoplasm. And what is really neat about this is that the conformational states of this repressor protein are controlled by lactose. So if lactose is present, it will bind on to the repressor, causing a conformational change that makes it release the operator. Whereas if lactose is absent, then the repressor is going to be bound on here. So if you have high levels, high concentrations of lactose, most of the repressor is going to be off of the operator, whereas if you have low concentrations of lactose, then the repressor is going to be bound to the operator. And that totally makes sense because if lactose is present, then you want to express these genes. However, if lactose is absent, then it would be a total waste of energy to express LAC-Z and LAC-Y and those other genes here. So when lactose levels are low, this repressor binds on and it prevents expression from taking place. So this is an example of what is called negative control because the binding of the repressor inhibits gene expression. Okay, now one thing that I think is sometimes useful for students to think about as they're learning this is this important point, that this repressor protein, it can only do two things. It can bind lactose or it can bind to the operator. Okay, it can bind lactose or it can bind the operator. And it has to do one of those two things. So it must bind lactose or the operator. So if you think about that, you can always check yourself, okay, if lactose is present, what state is the repressor going to be in? Well, if lactose is present, it's going to be bound to lactose. And in that state, it cannot bind to the operator, and therefore it cannot inhibit expression. Now, quickly again, going back to our logic of the Lac Operon, the behavior of that repressor protein really is responsible for these first two things that we see here, is how it ties in the expression of the Lac Operon to this these insights about lactose. So remember, the genes encoding enzymes that metabolize the lactose are only needed when lactose is present. And the repressor protein here, that repressor protein is what is really controlling the expression of the Lac Operon in the presence or absence of lactose. Now we're going to look at item number 3 here, which is where we start working lactose, glucose into the picture as well as lactose. Now let's go back to this graph that we had a minute ago. So in this graph here, the only sugar that was present is lactose. So in other words, there was no glucose around. So for this first line that we draw, there is no glucose. So let's think about how this would look different if there was glucose present. So I'm going to use a dotted line to represent that. So first of all, we're still looking at the expression of this enzyme, the beta-galactosidase enzyme, the expression of the LAC-Z gene. And so really there would be no difference here in the presence or absence of glucose for this. It would still be very low. Now here at this point we add lactose, and what you're going to see is you will get a little bit of an increase in expression, but it won't be very much, right? So not nearly as much as if you only have lactose present. And its dynamics are going to be the same, that once the lactose is removed, then the levels go back down close to zero. So this dotted line here represents what happens if glucose is present. So remember that glucose is the preferred energy source. And so this makes sense here too, right? If glucose and lactose are both present, you do get a little bit of expression of these genes, but it's not nearly as much as if lactose is the only sugar. So now we're going to look at how glucose influences the regulation of LAC-Z. So the influence of glucose is what we refer to as positive control, and you'll see how this differs for negative control in just a minute. Now, I'm going to, again, draw this region of the DNA where we have these regulatory sequences. Over here, again, is the start of transcription, so here's the information for the LAC-Z gene down here. Here we have the LAC-O, the operator region, and here I'm going to give myself a little extra room to draw. Here is this region called LAC-P, the promoter. Okay, so there is another protein that interacts with the DNA. In some ways it's similar to the repressor protein, which binds on here at the operator region, but this is a protein called the CAP protein, and that stands for catabolite activator protein, or CAP. And the CAP protein can bind here onto the promoter sequence. So again, it recognizes and binds to the DNA in this region. And it can also come away from the DNA. So we can show the CAP protein either binding on to the DNA or binding off. Now, if you remember, what happens when the repressor binds on here to the LAC-O is that it inhibits RNA polymerase from transcribing this gene. But the CAP protein, it actually has a positive effect. It makes that binding of RNA polymerase and the sigma factor to the promoter stronger. So in other words, you get a much stronger interaction here between RNA polymerase and the promoter. And of course, we also have to work in our little sigma factor here, this other protein that's involved. So when the CAP is bound, RNA polymerase, its binding to the promoter is enhanced, and that's why we call this positive control. Binding of the CAP protein enhances RNA polymerase binding, and therefore it also enhances expression of LAC-Z gene, of LAC-Z and the other genes in the operon. So also LAC-Z expression. Both of those get enhanced by having that RNA polymerase bound back on there better. All right, so what is it that controls the CAP protein? Well, this is where glucose works into it. So remember that when glucose is present, then LAC-Z is expressed but at a lower level, and that's actually consistent with the CAP protein being off. And so if you have high levels of glucose, in the cell, then the CAP protein is going to be off, and you're going to reduce the overall levels of expression of LAC-Z. Whereas if you have low levels of glucose in the cell, then the CAP protein will be bound on here, and the RNA polymerase will bind on nice and tight to the promoter, and you will get higher levels of expression of LAC-Z. But here's where things get a little tricky. If you remember with the repressor directly bound to the molecule that controlled it, the lactose. That's not quite so easy here in the case of glucose and the CAP protein. The CAP protein, in fact, binds onto another small molecule, which we'll draw on here, called cyclic AMP. We just abbreviate it little c AMP. That's cyclic AMP. And cyclic AMP levels are inversely proportional to glucose, and I'll show that in another drawing in just a second. Okay, so when cyclic AMP is bound onto the cap protein, then it will bind onto the promoter. If the cyclic AMP is absent, then the cap protein will come off of the promoter. So we'll come back to this drawing in a second, but let's think about how the cap protein works. There's, how the cyclic AMP works. So you're very familiar with this, ATP. And ATP can get converted into an interesting molecule called cyclic AMP, adenosine monophosphate. So 2 of the phosphates are gone. We can add those on here, 2 inorganic phosphates. And the remaining molecule gets, bonds formed to make it into a circle. And you can see images of that in the textbook. But the enzyme that catalyzes this here is called adenylate. Here's the enzyme that catalyzes this conversion. It's called adenylate cyclase. All right, so adenylate cyclase catalyzes the reaction of ATP and converts it into cyclic AMP and 2 inorganic phosphates. And this molecule here, adenylate cyclase, is inhibited by glucose. This is a good example of an allosteric interaction. The glucose will bind on here to the enzyme somewhere other than its active site and cause a conformational change that prevents it from doing its job. So, if levels of glucose are low, then you're going to make cyclic AMP. If levels of glucose are high, then you inhibit the enzyme and you prevent this step from happening. So now we can go back to our previous diagram here and add this in. So low levels of glucose, these correspond to high levels of cyclic AMP. They're inversely proportional, whereas high levels of glucose correspond to low levels of cyclic AMP. So that part's a little complicated and you have to keep all that straight, but the important thing to remember here is that this is an example of positive control. In the absence of glucose, this cap protein binds onto the promoter and allows this really tight interaction between RNA polymerase and the promoter, and that enhances the expression of the LAC-Z gene. All right, and so that explains, getting back to our graph, why in the presence of glucose you do get some expression. So this says that even without the cap protein bound on there to the DNA, RNA polymerase will still bind to the promoter and you can still get some expression, but it's not nearly the high levels of expression that you get in the absence of glucose. And finally, that gets us back to thinking about the logic of the Lac Operon here. So just to go over these again, if those genes that encode the enzymes to metabolize lactose, they're only needed when lactose is present, and they are expressed at very low levels when lactose is absent, and that's because of the binding of this little repressor protein. Now, if lactose and glucose are both present, then glucose is the preferred energy source, and we now know that the way glucose controls this is that in the absence of glucose, you don't have the cap protein bound, and without the cap protein bound, you can't get really high levels of the expression of these genes. So now what we're going to do is I'm going to give you a minute to think about some of these things, and the way we're going to think about it is to think about the effects of mutations, right? So mutations are changes to DNA sequence, and this is going to be important. I think it's a good way to review the way the Lac Operon works, but it's also going to be important for the lab that you're going to do related to the Lac Operon, where you're really going to work with different mutants that have changes to maybe sequences like the promoter or the operator. And you're going to ask, how do these DNA sequence changes affect the expression of the genes? And not just all of them, just the beta-galactosidase gene. That's the one we're really going to be focusing on. All right, so we're thinking about mutations, changes to all these DNA sequences and how they can affect the normal regulation of these genes. So this is a worksheet that you have in your book. And I'm going to set you up quickly, and then I'm going to ask you to pause this video and work through these answers. This is taken from an old exam, and then we'll go over them together. All right, so it says in wild-type bacteria, the LAC-Z gene is only expressed when lactose is present. So LAC-Z only expressed when lactose is present. This is what we're going to call normal expression. So if lactose is there, the gene is expressed. If lactose is absent, the gene is not expressed. So indicate how the mutations listed below would affect the expression of LAC-Z. In other words, so I'm asking you if we have a particular change, in this case, loss of function of the repressor. And let's just make this a little simpler by saying no repressor present. No repressor in the cell. It just doesn't have the information for making that particular protein. So no repressor in the cell. How would that change the expression? Would it have no effect? In other words, you would still get the same normal expression? Or would the LAC-Z gene now never be expressed? Or would the LAC-Z gene always be expressed? All right, so think about that. Here you have three different scenarios, three different mutations. Think of them individually. And also you'll notice that this only deals with the expression in response to lactose. It doesn't deal at all with expression in response to glucose. So pause the video now, try and answer these three, and then start it up again when you're ready to check your answers. Okay, I'm going to label these 1, 2, and 3 so we can start discussing them here. So with number 1, what's going to happen here if you don't have a repressor protein? All right. Well, what's going to happen then is that the LAC-Z gene is going to be always expressed. So normally it would only be expressed in the presence of lactose. But if you don't have the repressor protein, there's nothing there to inhibit that expression, right? So the repressor protein, in the absence of lactose, it's going to bind on here to the operator, and it's going to prevent RNA polymerase from reaching the start of transcription. But if you don't have a repressor protein, then there's never going to be anything bound here to the operator, and you're always going to be able to express those genes. So here's what's happening in number 1, is that there is nothing bound to the operator. LAC-O. Another way to say that is that there is no negative regulation possible. Okay, so how about for number 2? Deletion of the promoter, LAC-P. Well, LAC-P, as you'll remember here, this is the place where RNA polymerase first begins its interaction with the DNA. This is the initial point where RNA polymerase starts interacting with these genes. And if this sequence, the promoter sequence, is deleted or changed, then that's not going to happen. And so if you don't get any binding of the RNA polymerase to the DNA, you will never get the expression of LAC-Z. So in other words, for number 2 here, there is no RNA polymerase binding and so even though there may be lactose present and the LAC-Z gene is actually needed to metabolize that lactose, if there's no promoter sequence, then RNA polymerase is never going to express that gene. Now finally here, the last one, the deletion of the operator sequence. So here we're saying that everything that you see here is still present except that this operator sequence is now missing. Well, this is going to have a very similar effect to number 1, and that is that LAC-Z is always going to be expressed, because it's the repressor and the operator that interact together to inhibit the expression in the absence of lactose. So in the case of number 3, the explanation here is that there is now nowhere for the repressor protein to bind. And again, because of that, you're going to have no negative regulation. Now, we haven't really talked about positive regulation here with this worksheet because we're only focusing on the lactose side of the picture. But you could imagine a similar question like this that asked you to think about how the changes would be different if you had mutations in the DNA, and we were asking about conditions in which glucose is present. All right, so I just want to go back to this one more time and remind you that this is really important, thinking about this logic of the Lac Operon, because if you really keep these 3 points in your mind as you're studying all of this stuff, I think it helps, it helps it make sense. And the other thing you need to just think about is this part of the picture up here. What exactly does RNA polymerase do? It starts here at the promoter and then has to move across this operator region to get to the point where transcription starts. And if you really keep those things in your head, then it gives you a framework for really thinking about the positive and the negative regulation that you see in the presence or absence of both lactose and glucose. All right, good luck, and come to office hours and discuss this if you have any questions.